Dear students, I'm going to do now the production of indole that you will find on page 125, 126. That's where you're going to find it, 127 as well. But before I'm going to start there, I just want to do a little bit of revision on the direct microscopic count that is in lecture 6. And in lecture 6, you will find that we're talking about the microscope factor. The microscope factor is A times B over pi r squared. Pi r squared is a surface area of one microscope field. Okay, A is the surface area of the stain on the slide in square millimeters. Okay. And B is the reciprocal. And just to refresh your minds, 0.01 is the same as 1 over 100. To get the reciprocal of 1 over 100, reciprocal, the answer must always be 1. So 1 over 100, how am I going to get it to 1? 1 over 100 multiply with 100 over 1. So that 100 over 1, that 100 is your reciprocal. And the same, I said into that lecture, what is the reciprocal of 0.02 mol? Of 0.02 mol? Yes. 0.02 mol is 2 over 100. To get to 1, I must multiply with 100 over 2. Okay, so 2 goes into himself 1, and 2 goes into 150. The same on the other side. 2 goes into 150, and 2 goes in himself 1. So that one that is circled there, that is your reciprocal. So that reciprocal will be then 50. Another thing that I also just want to tell you, when we're doing anaerobic growth, it is important to know, is that anaerobic jar? Is it under anaerobic conditions? Is that glove box chamber? Is it under anaerobic conditions? How will we do that? How will we do that? No, very simple. Methylene blue, the stain methylene blue that we are using to stain bacteria, that stain is in the presence of oxygen, it is blue. In the absence of oxygen, it is colorless. So what are we doing? We take a piece of filter paper, we saturated it with methylene blue, that's a blue color, and if your anaerobic jar was under anaerobic conditions, it will turn, that piece of filter paper will then turn to a colorless color. And when you open it and you take that filter paper out, it will turn immediately blue in the presence of oxygen. Okay, I said I'm going to do production of indole. Now, let's see what it is. We want to see if the bacteria can produce indole on page 125. Amino acids are produced as a result of hydrolytic degradation of proteins, peptones, and peptides. You must know that. One of these amino acids, tryptophan, serves as a substrate for biochemical differentiation or characterization, since some species are capable of hydrolyzing this amino acid, and they hydrolyze this tryptophan to indole. So that is quite important. How do we see this process? One of the end products of this reaction is indole, which can be detected by a colorimetric test. This indole test is considered to be a very important test in the identification of organisms, because some organisms is indole positive, other organisms is indole negative. So that's normally one of the, when we're doing identification of organisms, that's one of the things that they always ask. Is the organism indole positive or is the organism indole negative? Okay. The tryptophan in the test medium, and it's normally a broth, is supplied by the peptone, and the peptone is commercially preparation of peptone, and it's called actually tryptone. It's standing in your study guide. Since it's only partially hydrolyzed proteins and contains a high concentration of tryptophan, which is degraded by the preparation procedures of some other kinds of peptone. Now, tryptophan is now being catalyzed by bacterial cells, not certain bacterial cells, certain bacteria groups, by the enzyme tryptophanase into 3 end products. And they show you the structure on page 125. They show you the structure of tryptophan being degraded by tryptophanase into 3 end products: indole, pyruvic acid, and ammonia. You must know this reaction and you must know the structure. So one of the end products now is indole. On the right hand side of page 125, they say there, indole production is detected by Kovacs reagent. Kovacs reagent is a yellow reagent. And the full name, the scientific name of it is actually para-dimethyl-aminobenzaldehyde, DMAB, and it is standing there on page 125 on the right hand side, and you must know it is para-dimethyl-aminobenzaldehyde, and as I said, it's a yellow color. Now, when we mix the indole in that nutrient broth containing now the tryptophan, and the tryptophan was converted by the bacteria to indole, and we mix now that mixture, that indole inside there, with the Kovacs reagent, it forms rose indole dye. And they show you that structure and reactions, and you must know them on page 125. And that rose indole dye is a red color. When we add this Kovacs reagent, it doesn't matter if it's 0.5, 0.6 mol, 0.4 mol, and you give it a good shake, the test tube, you will see it separates immediately. The Kovacs is going to lie on the top of the tube. And that top extracted the red, the rose indole dye, that's giving the red color. The rest of that medium will stay a brownish color. So they say in Kovacs reagent, the acidified DMAB is dissolved in amyl alcohol. And that amyl alcohol, when mixed thoroughly with the culture medium, the amyl alcohol extracts the indole but not the tryptophan. And the tryptophan is left in the rest of the test tube, and otherwise would interfere with the test tube, because it also reacts with DMAB. So for that reason, only the top part will be red. So after, this is a reaction that is taking very, very quickly place. So when the Kovacs on top is a red color, we say the organism is indole positive. When it's a yellow color on top, we say it is indole negative. On page 127, they say add 1 mol of Kovacs reagent to the test tube. No, if it's 1 mol, 0.5 mol, it doesn't matter. Okay, students, I hope that you understand the production of indole.